Assalamu alaikum. I am Dr. Saima Muzammil from Department of Microbiology, GCQF. Today I got the opportunity to present my work in third international conference on applied zoology. The research topic entitled is Biosynthesis of ZnO nanoparticle using Bacillus subtilis, characterization and nutritive significance from promoting plant growth in Siamese elk. Zinc is a micronutrient that is required in very small quantity, but it is very important for growth of the plants, animal, and human. Zinc deficiency caused a great economic loss in agriculture sector as growth of the plants are affected by zinc deficiency. There are many strategies to remedy the zinc deficiency, and one of them is the use of the nanoparticles. Nanoparticles are very small size nanoparticle, almost they are less than in 100 nanometer in diameter, and the use of the zinc oxide nanoparticle can be possible alternative for the zinc deficiency in the plants. They are very effective due to their small size, as they can be taken up by the plants more quickly as compared to the bulk zinc fertilizers. Thus, the use of the zinc oxide nanoparticle can be hypothesized to enhance the rate and to remedy the zinc deficiency in maize crop. There are different shapes and size of the nanoparticles that are responsible for their unique characteristics, and there are lots of methods to synthesize the nanoparticle, as there is the physical synthesis, there is chemical synthesis, and biological synthesis. In the biological synthesis, use of the microorganism is a major field, and in the other side, plant tissue and different plant extract can also be used for the synthesis of the nanoparticles. As biological synthesis is eco-friendly as compared to the chemical and physical synthesis, in our study we use the biological synthesis of uh, zinc oxide nanoparticle, and then we characterize the zinc oxide nanoparticle, and after all we check its the response of the maize crop to the nanoparticles. For this one, the seeds of the maize crop were primed at different concentration of zinc oxide nanoparticle at 2 mg per liter, 4, 8, and 60 mg per liter. And afterward, we determine the growth biomarkers, that is, we determine shoot and root length, we determine the shoot and root fresh mass and dry mass and the leaf area of the plant to check the effects of the nanoparticle on the plants. So this is the after synthesis of the nanoparticle. This UV spectrometry shows the peak at 331 nanometer, which corresponds to the biological synthesized zinc oxide nanoparticle. And here we see the same micrograph of the zinc oxide nanoparticle, and this is the XRD of the nanoparticle, and these peaks shows the elemental structure of the zinc at the nano size. Afterward, the TEM size was calculated by TEM micrograph. Here we show in this picture we can see the spherical shape of the nanoparticle, and the size was calculated according to this. The average size was 16 to 20 nanometer of these nanoparticles. This is the XPS analysis of the nanoparticle, and here we can see the elemental form of the nanoparticle, and these graph showed the synthesis of the zinc oxide nanoparticle. This is the size distribution by number and zeta potential of these nanoparticles. After all this characterization, we check these nanoparticles on the plants, and different growth parameters were determined, and here in this picture we see that after priming with the different concentration of the nanoparticle, the root length and shoot length of the plant has increased as compared to the control, and same is the case with the shoot dry mass, root dry mass, and fresh and shoot and root fresh mass. But here we notice that the significant enhancement has been observed as compared to the control in the 8 mg per liter concentration. And after that concentration at 16, a decrease in the root and shoot length and the fresh and dry mass has been observed. Same is the case with the leaf area and total protein content of the plant. Here in this picture, we see the leaf area that is maximum at 8 mg per liter of the nanoparticle, and protein content was also more here at this concentration. Maybe this is the reason that at higher concentration, the zinc nanoparticle, zinc oxide nanoparticle, they have the negative impact, as the more concentration than the required could reduce the growth of the plant also. So in the end, our conclusion is that zinc oxide nanoparticle can be effectively used as zinc source to overcome the zinc deficiency in maize plant. And nanopriming could be an efficient technique for the better production of the crop. So zinc can be used as nanoscale. That is a revolutionary step in the agriculture. Thank you.